Good morning, everyone. My name is Alexandrov, and I'm an associate professor at UC San Diego. And what I'm going to tell you today is how one can use cancer mutational signatures to explore the etiology of cancers with a specific focus on pediatric cancers. I'm going to start my presentation by giving you an overview of what mutational signatures are. And I'll start by just describing somatic mutations. Somatic mutations accumulate daily in every single cell of the human body. These mutations can accumulate from lifestyle choices, from failure of cellular machinery, or even from the normal operations of the cells. We have known for quite a while that cancer can be strongly affected by somatic mutagenesis. The poster child example for that would be tobacco smoking, with smoking generating many mutations in many different organs, and probably the most prominent being the lung. And indeed, one knows that if you look at lung squamous cell carcinoma, one of every 105 patients with this adulthood cancer will be smokers, and only one will be a never smoker. So if there wasn't this lifestyle choice, this cancer type will be almost non-existent. Now, we know that cancer risk can be affected by somatic mutations, but can we use somatic mutations to understand the processes that actually cause the cancer? And indeed, one of the approaches that exists for that is called mutational signatures. This is a machine learning approach that allows to detect unusual patterns of mutations. And then one can associate these unusual patterns of mutations with different mutagenic processes, being these processes within the cell or being lifestyle choices or environmental exposures. I just want to give you some examples from adult cancer. So if you look at skin cancer, most of the time one would associate that with ultraviolet light. And indeed, the majority of ultraviolet light associated skin cancer will bear a very specific pattern of mutations. This is predominantly C2T mutations and dipyrimidines, and you can see here the actual pattern of the ultraviolet light mutational signature. If one looks at lung cancer of tobacco smokers, approximately 80% of all types of lung cancer are caused by tobacco smoking. And again, when we look at the cancers of these patients, you're going to see very specific patterns of mutations, very characteristic molecular fingerprint, and indeed, this can be very well reproduced in experimental studies. During the last probably decade, we and others have created a compendium of these mutational signatures across the spectrum of human cancers, and we have found over 100 of these signatures, some of them shown here. We have signatures associated with endogenous processes, whether they're being normally operating or defective. We have treatment-associated signatures, and we have environmental mutagens and lifestyle choice-associated signatures. So how can one use these signatures to investigate unusual patterns of cancers? And I'm going to show you two examples, one being in adult cancers and one being in childhood cancers. So we first generated the first reference set or the first compendium of mutational signatures in 2013. We had about 21 of those signatures, and we did a refresh in 2015 when we discovered another 9, and we described the signature called signature 30, which has unknown etiology. And we reported it, we published it, and we left it there. Another group from the Netherlands took that and was able to show what causes this specific signature. And indeed, if you have failure of a specific gene, NTHL1, that results in failure of DNA repair, basic excision repair, and it will cause this specific pattern of somatic mutation. So now you have a mechanism behind that. And if one fast forwards 2 years afterwards, actually using this signature as a phenotype, the signature can be used as a multi-tumor phenotype for detecting germline predispositions. And indeed, one can start screening patients and seeing whether they're predisposed of actually getting these cancers because they carry these germline variants. And again, this specific example is for adulthood cancers, but I also want to give an example for childhood cancers, which is a very striking example of ultraviolet light. And this is a study led by Jinghui Zhang, who is also speaking in this symposium. And they're looking at a number of leukemias. We started seeing actually the signature of ultraviolet light. And again, this is the pattern of the ultraviolet light signature, and I'll show you how these samples look, and you can see that they're remarkably similar. And this is the combined. We can zoom in into different individual samples, and you can see they bear exactly the same mutational signature. And what that tells you is actually UV light is mutating these B cells, and it's very likely causing this specific leukemia in children. One can do an extended analysis to a number of additional features. They would all match. This could be confirmed now a number of times. And also, it has actually been shown to occur only in white Caucasian children. And that means that exposure to UV light in these young Caucasian children can increase their risk for developing this specific type of leukemia. And indeed, there has been even an epidemiological confirmation from a large-scale French epidemiological study showing that UV light is an environmental risk for this specific type of lymphoblastic leukemias. So these were my examples, but how can we use mutational signatures to investigate the unusual patterns of pediatric cancers? And what I'm going to show you is a recent analysis, a recent collaborative analysis led by the German team, Natalie Jager and Stefan Pfister, which examined 785 childhood cancers from many, many different types. And essentially, they mapped the mutational signatures of the different processes causing mutations in these cancer types. And the first thing here, you see a table, you see the different set of known signatures on the left. They have specific known etiologies. And then the circle tells you what proportions of samples of a specific childhood cancer type have that signature. And the color tells you how potent it is, how many mutations does it generate in an average sample. The first thing you'll see in this figure is very sparse. There are very, very few mutational signatures in childhood cancers. If you look in the alternative figure for adulthood cancers, it will be overwhelmed of different circles in different sizes across all different cancer types. We can briefly categorize these processes. We have the clock-like mutational process. These are processes that tick in every single cell of the body. They occur just because the cell exists. They're very, very prevalent in both childhood and adulthood cancers. We have processes just because something failed, whether it's a failure of DNA repair, or whether it's a failure of a specific proofreading domain of a polymerase, or whether there is an infidelity of a specific polymerase. They can generate different signatures, and they can actually result in specific mutations that cause cancer. When it comes to mutational signatures, we can also detect signatures of prior therapy. And when we look at some of those cancers, indeed, we can see that patients who have been previously treated for either another malignancies or for have gotten a secondary cancer, we can actually see the imprint of that treatment. And indeed, in some cases, we can show that the prior treatment causes the secondary cancer. The only environmental factor that we can find with mutational signatures in this study is the one of UV light in B-cell ALL. And again, that's a completely different cohort than the one I previously showed you. And as a matter of fact, we haven't found any other environmental or lifestyle choice based on mutational signatures. The other signatures of interest is the Apobec family of deaminases. These are enzymes that can be activated by viruses. In adulthood cancers, if you have an HPV infection, they'll get activated very strongly. They can get activated by inflammation. They can get activated by other endogenous and exogenous mechanisms. There is no evidence here that they're getting activated by external factors in these childhood cancers, but it's possible. The mutational signature for reactive oxygen species is very prevalent of childhood cancers, much more than in adulthood cancers, and we do not understand why that's the case. And in addition to that, there is a number of signatures that are with unknown etiologies, and it's entirely possible that they're caused by lifestyle choices or environmental exposures. So in summary, I hope I was able to show you that mutational signatures is an approach that allows you to detect the etiologies of cancers from genomics data. In adulthood cancers, we have many, many examples of signatures associated with environmental mutagens and lifestyle choices. In pediatric cancers, that's not the case. The majority of signatures are due to endogenous process or failure of these processes. And if one starts looking for environmental exposure or lifestyle choices using mutational signatures, there is the very clear example of UV light in B-cell ALLs. And there is potentially the Apobec-associated signatures and the mutational signatures of unknown etiologies. Thank you for your time, and I'm looking forward to the panel discussion.